And now I'd like to introduce you to Patrick Turley, who is a mathematician and economist by training. He's also an instructor at the Analytic and Translational Genetics Unit at the Massachusetts General Hospital and many more prestigious institutions. And his research fields include social science, genomics, statistical genomics, econometrics, and economics of education. And the floor is yours, Patrick.
Great. Hi, everyone. Someone stop me if you can't hear me, but so I'm going to talk today about assortative mating and using polygenic scores to estimate assortative mating, even if you have samples of unrelated individuals. So you don't have any information on parents or spouse pairs. Oh, wait, it's not advancing. Okay, great. First, what is assortative mating? Well, just as at a high level, assortative mating is just the factors that cause parents to be mating together and producing offspring. And so you might imagine in a random mating population, there's no rhyme or reason determining who is paired with whom. However, if you have a population that's mating perfectly, then you might see people who have similar phenotypes having children together. So sorting can happen within phenotypes or across phenotypes. So what I mean by that is you may have that tall people tend to have children with other tall people. So that would be within a phenotype. But you also could imagine that there's sorting across phenotypes. So, educated people may be more likely to have children with tall people. So we're going to consider both of those things. There's been a lot of work looking at assortative mating in human and non-human populations, but assortative mating in human populations has been widely documented in a lot of different papers. Today I'm going to talk about sorting specifically on polygenic scores. So I appreciate the previous talks that have described a little bit what polygenic scores are, but specifically I want to know, do people who have a high polygenic score for a particular trait tend to be matched with people who also have a high polygenic score for that phenotype? In general, it's very difficult to estimate assortative mating and the primary reason is that it takes a lot of data to produce these estimates of assortative mating. We just don't have large samples of parent pairs that are publicly available. I mean, more and more we're starting to see those, but currently it's very difficult to get large samples like that. So what we're going to do is we've developed an approach that allows you to infer what the correlation was of the polygenic scores between parents using only data from their children. Because the children inherited their DNA from their parents, there is some information there about how similar their parents were. So just quickly, why should we care about assortative mating at all? Well, turns out that for heritable traits, if there's assortative mating on that trait, it leads to an increased severity and prevalence of disease. It can reflect changes in migration and social preferences. And so if people are sorting geographically or based on education or other kind of factors like that. Assortative mating creates bias in genetic studies. And so, it potentially inflates GWAS summary statistics, can affect Mendelian randomization studies, all the type of genetic studies that maybe we're interested in. Assortative mating can create bias in those things. It also drives genetic correlations between traits. And so if there's been a lot of work trying to measure how genetically correlated different sets of traits are. And it would be important to know whether or not that correlation is being driven based on common biological factors, or if it's just a result of assortative mating. So the intuition, just at a high level behind the approach that we're going to use, is that assortative mating inflates the variance of polygenic scores in a predictable way. And so if we look at polygenic scores and we see how much the variance is inflated, then we can use that to infer how much assortative mating there must have been in the parent's generation. So to describe a bit how this works, the key principle is that assortative mating increases long-range LD in in the population. And so let me describe how that works. So pretend that we have two SNPs, A and B, and both of these SNPs increase height, but they're on different chromosomes. So traditionally, in a randomly mating population, we would expect that these two SNPs, the genotypes, to be uncorrelated with each other because they're inherited independently. But in a sorting population, imagine that you have a child who has allele A. Well, if they have allele A, then they got that from somewhere, so their parent must have also had a copy of allele A. And if the parent has a copy of allele A, that means that that parent is taller, because we said that allele A increases height. Well, if parent 1 is taller and there's assortative mating, then parent 2 must also be taller on average. And if parent 2 tends to be taller on average, then it means that they're more likely to have every allele that increases height, including this allele B that we also talked about. And so if parent 2 has a copy of allele B, then their child is also more likely to have a copy of allele B. And so through this chain, we see that if a child has a copy of allele A and they're sorting in the population, this causes allele A and allele B to be correlated with one another, even though they're on separate chromosomes. So what implication does this have on polygenic scores? Well, imagine that we're calculating what the variance of some polygenic score is. Well, a polygenic score is just a weighted sum of genotypes, like Barbara explained to us. And so if you take the variance of this sum, then you have a sum of all these variance terms, but then there's all of these covariance terms as well. And so these covariance terms is the correlation between every pair of SNPs in the genome. Well, in a randomly mating population, most SNP pairs have a correlation of exactly 0, like especially when they're on separate chromosomes. And so since the vast majority of SNP pairs are cross-chromosome SNP pairs, this term is largely a bunch of zeros. However, if you have a randomly mating population, all of a sudden you have positive covariance between all of your SNPs. And so this causes the variance of your polygenic score to push outward. And so this is the sense in which I said random assortative mating increases the variance of polygenic scores. So I'm not going to go deep into the specific estimation strategy for this method, just for the sake of time, but I am going to just give it a high level features of the estimation strategy that we're going to use. So first off, our method estimates the correlation between parental polygenic scores, just like we want. So you have some data, and what you would be estimating with the data is the effect, the correlation between the polygenic scores of their parents on average. Another nice feature of this method is we can measure assortative mating over time. And so it doesn't just give you a single number of how much sorting there is, but it'll give you a non-parametric curve about what's been happening to assortative mating over the time frame that you have data. And so this will be useful if we're wanting to think about trends in sorting in history. As I said before, this doesn't require data on parent pairs, and so this is going to be really powerful from a data standpoint, because we could use data sets like the UK Biobank, which has been discussed previously in this session, which is just an enormous number of individuals, but not necessarily individuals who are linked to parents, or parents who are linked to each other. Another important feature of the method is that it's robust to population stratification and even ascertainment under somewhat weak assumptions. And so if you're worried that the results are just being driven by your funny sample selection algorithm like you have in the UK Biobank, or if you think it's driven by just stratified populations, under some weak assumptions, we can at least rule those out by the method that we've developed. So I'm going to now show you a bunch of results applying the method that we developed to a number of phenotypes, to show you the kinds of lessons that you can learn from applying our method and learning about assortative mating. We're going to first create polygenic score weights from GWAS summary statistics. using the methods that that Barbara described. In general, these summary statistics that we used are either publicly available or we produce them in-house, just doing a GWAS in a subset of the UK Biobank. And so then the results that I'm actually presenting are estimated in a subset of the UK Biobank, specifically in the European ancestry subsample of the UK Biobank, which means that we're able to estimate assortative mating in a sample of about 200,000 individuals, which is much bigger than any data set that I know of that links parent pairs. Okay, so I'm going to start out with an example which I think is a bit unintuitive, but also highlights how this method is robust to population stratification, and this is latitude. So what we did here is we took half of the data in the UK Biobank and we did a GWAS on a person's reported latitude of birth. When we did this GWAS, we didn't control for principal components, and so you should think of these results as nearly 100% population stratification. Nevertheless, it may still be interesting because this polygenic score that you create from such a GWAS is a predictor of where a person's from, even if we shouldn't think of that predictor as some causal, through a causal mechanism. And so we have this polygenic score for latitude. We run it through our method and it produces a figure like this one that I showed here. So to explain how to read this, on the x-axis, we have year of birth of the individuals. And so what this year of birth of the individuals, on the y-axis, I have this measure M of T. So M is the correlation of the polygenic scores of parents who are having children in year T. And I think that we actually see something kind of interesting here in this figure. So first off, we see kind of early in our data, we have moderately high correlation of latitude, meaning that people are sorting geographically, just as you'd expect maybe in the late to mid 30s. But then very quickly, there's a sudden decay in the correlation, and there's very little sorting in the years right around World War II and immediately after World War II. And I think this is representative of the geographic, there were lots of people moving around during this wartime period. But then shortly as the UK is recovering from the war, we see that sorting is increasing again. People are going back to where they're from and not moving around within the UK very strongly. And so I mean, I think you can tell kind of a fun story just using an unconventional polygenic score. I claimed earlier, however, that that this method is robust to population stratification. And this example that I gave you here is 100% driven by stratification. But if we do the stratification correction of this method, we see that these potentially interesting trends in our first graph entirely disappear. And so we get approximately 0 in all years studied. And so I think that should give us additional confidence that the next few examples that I show you, all which have the population stratification correction, that those are driven actually by sorting on some sort of related phenotype and not population stratification within the UK. So the first two examples that I want to talk through are two phenotypes where it's known that there is a high level of sorting already phenotypically in the population. In fact, so this is height and education. And there's been a lot of work, and in fact, these are the two phenotypes that almost always show the highest degree of sorting in any phenotypic study. And so, similarly, we see things here. So again, on the x-axis, we have year of birth of the children, and on the y-axis is the correlation of the polygenic scores of their parents. And so, we see moderate correlations here, about 15% overall, maybe a tiny bit higher for educational attainment. But one thing that I think is interesting that you can learn from this figure is not only is there a moderate amount of sorting on polygenic scores for these individuals, but we see an increasing trend suggesting that if you look at the polygenic scores of individuals, of the parents of individuals who were born in 1970, that will be higher in those individuals than the people who were born in in the late 1930s, and the same for education. We see that individuals are more strongly sorting on their educational attainment polygenic score than they were in the early 30s. With some additional assumptions, you could then translate that into saying, well, people maybe are more likely to be sorting on the phenotype of height and the phenotype of educational attainment. And I think this is somewhat sensible, given the, more people going to college and people pairing up during the college years. And you might imagine that this would increase the amount of educational attainment sorting that you see in the population. The next two examples I want to give are two phenotypes where you see actually a high level of correlation between spouse pairs phenotypically, but in our model we estimate pretty low levels of correlation. So notice here the y-axis has changed. In both of these cases, it looks like maybe there are a few years that are positive, but only barely. And the largest it gets based on our confidence intervals is a correlation of 0.06, maybe 0.07. And so this might be interesting because it answers this question. I just noticed I misspelled lock. This is lock et al. But this answers the question of, we see a strong amount of phenotypic correlation between autism-related phenotypes and in terms of BMI of spouses. And you might wonder, is that because these individuals are pairing based on things that give additional risk for BMI or autism? Or is it that these individuals spend a lot of time together, and so at the point that you measure the BMI of a spouse pair, for example, and you take the correlation, this pair would have similar diets, maybe similar activity levels, and so they might have a similar BMI just due to their common environment over the years. I'm not an autism expert, and so I can't tell a similar story for autism spectrum disorder, but it does appear, at least based on this, that the correlation that's seen in autism-related phenotypes doesn't seem to be sorting on genetic autism risk, or at least the genetic autism risk that's captured by a polygenic score. So this has all been sorting within a trait. I want to talk about cross-trait assortative mating and why we might care about that. So it turns out that education and depressive symptoms are moderately highly genetically correlated. So they have a genetic correlation of -0.3, meaning that people who get more education tend to be more depressed, which I imagine is not surprising to many of the people on this call. But the question is this a causal relationship? Is it that there are common biological factors that cause a person to both go further in school and become depressed? Or does education cause depression, or does depression cause a person to go further in school? These are all, possible stories that might drive why we're interested in this genetic correlation. But another possibility that is less interesting is if it's just the case that there's assortative mating, and it's that this genetic correlation that we estimate is purely a result of sorting that's happening in the population. And so using this method, we can estimate what fraction of the genetic correlation between education and depressive symptoms is driven by sorting. And that's what's in this figure here. So again, on the x-axis, you have year of birth, and on the y-axis is the fraction of genetic correlation that's due to assortative mating. And we see that in the most recent years, this genetic correlation, almost a third or even over a third of the genetic correlation between education and depressive symptoms is purely due to assortative mating. And so this might temper any enthusiasm over research into trying to find the common cause that leads this relationship. So in summary, I've presented a method and some results that show that assortative mating can be reliably estimated in publicly available samples of unrelated individuals. If you just have large data sets like the UK Biobank or other data sets that are becoming more and more available, and then publicly available summary statistics as well. And that using these methods can reveal important insights about human behavior, help us interpret our genetic studies for disease, and and other phenotypes. And so, I hope that using these kinds of methods to think about genetic assortative mating will be useful and standard to whenever we're talking about relationships, genetic relationships between phenotypes. So of course, thanks to, this was a big project involving many people. I'd like to especially thank Michael Bennett, Rosie Lee, and Grant Goldman, who are 3 research assistants that I've worked with on this project. And so a lot of the hard work that has gone into this has been theirs. Thanks to funders and the conference organizers. And if you'd like to reach out to me later, up in the corner here, I have my website and email address, Twitter handle. So feel free to reach out. Thank you.
Yeah, thanks a lot, Patrick. So I also cannot answer your question about autism, although I think the more time you spend with nerds, the more nerdy it gets. So a provocative question about Dutch people. Why are they so large? Do they appreciate that feature more in assortative mating, or how can you test for that?
You're asking about Dutch people within the European population, or with...
I thought they are the largest population at all. So
yeah. So if you had data, from all over Europe, and so I'm trying to think of a large European data set. I mean, in theory, you could calculate how much of that is due to genetic assortative mating. I don't, I mean, I know there are lots of hypotheses about why the Dutch are the tallest. I'm not an expert on that. I think that there's a lot of both environmental and genetic factors that people attribute to it, though.
Okay, and Julian asks, if you select offspring based on affection status, e.g. cases only, gene-gene interactions or recessive effects could introduce similar correlations between parents. Did you look at something like this?
So the gene-gene interactions could induce a correlation between parents. So I mean, if you had a randomly mating population, it's not clear to me how you could have how that would induce correlation between parents that isn't there already. Perhaps I'm misunderstanding the question.
So I think that if you select samples based on the affection status, so basically that all offspring are cases.
Oh, yes.
Then there is a likely chance that if they are gene-gene interactions, that the parents match to have this because they are the parents of an affected offspring. So that
that's definitely true, and it's actually potentially even more sensitive. So I claimed earlier in the talk that the method is robust to ascertainment, and so this I think gets at the question that you're asking. So what I mean a bit more precisely is that if you can describe ascertainment as a linear function of the polygenic score, so if the likelihood of you being in the sample relative to the whole population is a linear function of the polygenic score, then our methods are robust. If your selection algorithm is that you oversample cases, though, I don't think, and you have a highly predictive polygenic score, then I don't think it would be linear, and so that may induce some bias into.
Yeah, so I know that you said it's robust, but I basically just mean it as a feature. So if you would look at cases only, you're not maybe interested in assortative mating, but in gene-by-gene interactions or recessive effects, that you could use your method to do something similar in cases only for a different purpose. So it's like a additional feature, maybe.
Oh, I see. Yeah. We rely pretty heavily on the additive nature of the polygenic scores that we use. So I think I think it certainly would be possible to tweak things to get at the kinds of things you're bringing up, but it's not clear to me that it would be like an immediate. You couldn't port it over directly.
Okay, thank you.
And we have yet another question from Maria. So considering your correlation between assortative mating with several traits, do you think in a longer time period, human genome would see lesser genetic heterogeneity causing rather reduced evolution?
Yeah, that's a good question. I mean, what the traits that I've considered here are very polygenic traits. And so as a result, the effect of, even if there's high levels of sorting on a very polygenic trait, you know, the actual effect on any particular SNP can sort of like, any SNP level effects tend to be really small. I mean, over a long amount of time, and you may worry about things like that. And there is some work by Augie Kong and another paper by Jonathan Beauchamp that look at these kinds of things and how sorting may affect you know, average levels of different types of polygenic scores in the population. You know, and I think that the numbers they found were like big enough that it was interesting, but not so big that I think we're going to start seeing an explosion. You know, big huge demographic changes in human evolution.
Okay, thanks a lot, Patrick, again.